In this lecture, we are going to discuss the bioinformatic tools for inferring evolutionary relationships between biological sequences, also called phylogenetics. What is phylogenetics? Phylogenetics is the area of research concerned with finding the genetic connections and relationship between species. The basic idea is to compare specific characters or features of the species under the natural assumption that the species with similar characters are genetically close. The term phylogeny refers to these relationships, usually present as a tree of relations or a phylogenetic tree. A phylogenetic tree is an history of lineages of organisms as they change through time. It implies that different species arise from previous forms and that all organisms from small microbes to large plants and animals are connected by the passage of genes along the branches of the phylogenetic tree that links all life forms. Let's take a look to this simple phylogenetic tree. From left to right, we have a sponge, a lobster, and a chimp. Lobsters and chimps are more closely related to each other than either all to the sponge. So, their lineages intersect first. Sponge, lobsters, and chimps are all animals. Thus, their lineages intersect at some point. Phylogeny also includes information of time, with the present at the top of the tips and the past at the bottom. In this way, the blue dot represents the last common ancestor of all animals, and the red the last common ancestors of all bilaterians. A phylogenetic tree is a structure in which species are arranged on branches that link them according to their relationship or evolutionary descendants. Classic phylogenetics deal mainly with physical or morphological features, like the size, color, number of legs, etc. Charles Darwin produced one of the first illustrations and crucially popularized the notion of an evolutionary tree in his seminal book, The Origin of Species. A typical rooted tree with scaled branches is illustrated in the figure and corresponds to an illustration of the book of Darwin. Over a century later, evolutionary biologists still use tree diagrams to depict evolution because such diagrams effectively convey the concept of speciation from the splitting of lineages. But in this case, their relationships are estimated by the comparison between nucleic acid or protein sequences. Evolution is a process whereby populations are altered over the time and may split into separate branches, hybridize together, or terminate by extinction. The evolutionary branching process may be depicted as a phylogenetic tree when you describe the evolutionary relationship between organisms, species, or even proteins or genes. Phylogenetics are a basic tool in the study of molecular evolution. They allow to reconstruct the history of successive difference between genes as well to obtain information of their ancestors. The phylogenetic tree reconstruction is a statistical problem. It is an estimation of real evolutionary events and should be tested for their accuracy. In practice, phylogenetic analysis tend to generate phylogenetic trees with accurate regions and less precise regions. The parts of phylogenetic trees include the tip of the tree branches represent the taxa in the study. Taxa may be at any taxonomic level, order, species, populations, proteins, genes, etc. These taxa may be called OUTs or operational taxonomic units. They are also called leaves. The lines within the tree are called the branches. The points at which branches connect, or the tip of branches, are both called nodes. Internal nodes connect branches. External nodes are the tips that represent the taxa. Some trees will have a basal node, known as the root. A grouping of an ancestor and all his descendants is known as a clade. Phylogenetic trees could be rooted or unrooted. Rooted trees has a single node that represents a point in time prior to any other node in the tree. They have directionality because their nodes are ordered by their appearance in old or recent. Unrooted trees, on the other hand, only specify the degree of relatance between the taxa, but not the evolutionary path. They have no directionality, so you cannot estimate what nodes are older or newer. Phylogenetic trees could be scaled or unscaled, also called ultrameric. The scaly trees, the length of the branches are proportional to the number of changes that occur in that branch. On unscaled or ultrameric trees, the branch lengths are not proportional to the number of the changes. All of them are at the same distance from the ancestral nodes. A monophyletic group is characterized by the inclusion of all descendants from a single common ancestor, whereas a paraphyletic group do not include all the descendants of a single common ancestor. This means that when the group has a common ancestor, we artificially ignore a subset of his descendants. A polytomy, meaning many temporal base branches, indicates that evolutionary relationships cannot be fully resolved to dichotomies. Dichotomies are branch points on a tree that has two immediate descendants. Polytomies could be caused by lack of a knowledge that normally means that there is an efficient data to understand how the lineages are related, or could be caused by the rapid speciation events in a population. Usually, we want to obtain a phylogenetic tree with no polytopes, which means a fully resolved tree. Finally, an important feature of phylogenetic trees is that the topology of a given tree not change if you make turning movements at the nodes of the tree. You can rotate the tree and the topology of the tree is not changing. This allows, once you have calculated a given tree, you can make rotation movements and nodes to organize the data for better visualization.